Wonderful. Again, so thank you all so much for joining us, and it is my pleasure to introduce our speaker today. Dr. Jessica Whitted is an assistant professor of stem cell and regenerative biology at Harvard University. After earning a BA in philosophy and a BS in biological sciences from the University of Missouri, she obtained her PhD in biology from MIT, where she studied in Paul Geraghty's lab. Dr. Whitted's PhD focused on molecular mechanisms controlling the development and maintenance of cellular architectures in the Drosophila nervous system. After her PhD, Dr. Whitted became a postdoc in the Tabin Lab in the Department of Genetics at Harvard Medical School. Now, Dr. Whitted runs her own lab in the Department of Stem Cell and Regenerative Biology at Harvard University. She and her team focus on limb regeneration in axolotl salamanders, developing tools to manipulate gene expression during limb regeneration. So please join me in welcoming Dr. Jessica Whitted.
Hi, thanks so much to the other Jessica and to Marty for inviting me. Really excited to be here. My high school and college teachers were amongst the most influential people in my life, and my mom is a teacher. And so it's special to be able to talk to teachers who want to bring the most advanced technology into their classrooms. So today I'm going to give you a super brief reminder about how we got to this place where humans can manipulate the expression of genes in other organisms. And today we're going to specifically focus on this idea that we can get rid of gene activity in other organisms so we can learn about what those genes normally do. But then we're going to also pivot to how we can use these kinds of technologies for medical purposes and also for purposes outside of that. So just to remind you, cells incur a lot of DNA damage on a regular basis. And there's lots of different targets in a DNA molecule for damage. So just shown here in the red, the green and the blue arrows are the kinds of places within these molecules that particular molecules can be deranged. So for example, there can be spontaneous oxidative damage, hydrolytic attacks, and methylation changes. And without getting into all the details about where these kinds of insults come from, we could ask probably even someone in elementary school or middle school, and they might be able to tell you why it's bad to go out in the sun without sunblock, or why it's bad to be around cigarette smoke. And both of these can eventually lead to DNA damage. And then finally, cells also incur DNA damage as a part of their normal existence. So for example, like during the process of DNA replication, DNA can become deranged. And so cells have evolved very sophisticated mechanisms for making sure that they can correct this kind of damage, because if it's not corrected, then the cell can go on to become cancer, amongst other things. And so I don't want you to read all the nitty-gritty details here, but what I want to draw your attention to is the frequency with which cells have to cope with these insults. And so if you look here, that this chart is basically telling us in a typical mammalian diploid cell, how many particular events might be incurred within 24 hours. And you can see that there are literally thousands of them for most of these things in any one cell. And so all I want you to appreciate here is that it's really important that these things get corrected, and that cells over time have evolved really intense mechanisms for ensuring that they're corrected. And if we look at these kinds of mechanisms, there's two that we need to think about today. And on the left, we have something called base excision repair, and on the right, something called nucleotide excision repair. And the way I like to think about this is that a base excision repair is kind of like a tiny defect, so like one particular base, like this kid here who is missing a front tooth. Whereas the nucleotide excision repair swoops in when there's been a bigger lesion, or when the only way to cope with the type of lesion is to remove a big stretch. And so here, if we get back into the kinds of mechanisms by which these things get corrected, some of the enzymes are common. And so there are special enzymes in the case of base excision repair that removes just the base, and that goes on to remove the sugar phosphate. And then the common thing is that the nucleotide, the correct one gets added by DNA polymerase, and then this nick in the backbone is sealed up by DNA ligase. And if we look at the bigger mistakes, you can see that here, first a big chunk has to get removed by a different kind of enzyme. And this one is called a particular enzyme that excises an entire piece of DNA. This is an excision nuclease, and it also involves an unwinding event by a DNA helicase. But then, just like the base excision repair, ultimately new nucleotides have to get filled in by DNA polymerase, and then the nick is sealed up by DNA ligase. And if we're thinking about the ways that double-strand breaks get repaired, Sometimes they get repaired in this way, which is called non-homologous end joining. And in this particular method, you can appreciate that some information is lost because basically the two ends get stuck together, right? After there's been some processing or elimination of genetic material. But today we also want to think primarily about this process of homologous recombination. And so during the process of homologous recombination, information from the sister chromatids are used as a template for filling in missing information in the DNA strand. And this looks a little bit more like this if we really get into the full glory. And you can see here that all I want you to see is that there's this homologous recombination event that I just highlighted right then and there, and it sort of goes up, and one strand can invade the other one. And the most important thing to see here is that this process is mediated by DNA base pairing itself. And the DNA genome editing technologies really exploit this feature of cells. And so you can see over here that eventually what happens is that there's this thing called strand invasion, and then eventually DNA synthesis does kick in, and the new DNA is made, and then it's ligated together, and ultimately you can get repair. So this is pretty cool because it is most famously the thing that happens during meiosis, whereby we can get new combinations of traits. So like, you know, if we just pretend in a really hypothetical situation that hair color is on the same chromosome as eye color, and we know this is totally hypothetical because both of those are controlled by many genes. But if we just say that these two things are on the same chromosome, and if sometimes you want to get a new configuration, like you want brown hair and blue eyes or blonde hair and brown eyes, then that can happen through homologous recombination. And so this is really what fuels differences in a population over evolutionary time. But what we can also think about it as a way that can be harnessed for people to really manipulate genomes. And so how can this happen? So first I'm going to highlight some of the earlier work that was done to make transgenic and knockout animals, specifically knockout animals is what we're going to focus on. And these technologies largely exploited these endogenous homologous recombination mechanisms that I just told you about. But then we're going to move on to these sort of next generation DNA editing technologies that are really based on the CRISPR-Cas9 system. And that is sort of what I was tasked with talking about today, but I wanted to give you a little bit of the history about how these kinds of editing technologies existed before people started exploiting CRISPR-Cas9. Okay, so one of the ways that people generally learn about what genes do in a mammalian context, and especially in disease, is that they try to model a phenomenon or a disease in a mouse. And so the mouse has really emerged in the last several decades as like the powerhouse of mammalian genetics, because you can go into a mouse and in a relatively short amount of time, you can make a customized mouse. So if we just pretend that the human has one particular gene and we think that in some people there might be a mutation in it and it renders it faulty and causes a disease, what we might want to do is identify that gene in the mouse and go in and, for example, edit it out, remove it, and see if you get the disease. Another thing that you might want to do is, instead of just removing the whole thing, maybe you want to mimic the mutation that the human patient has and see if you give the mouse that mutation, if it has the disease. So both of these kinds of operations can be achieved with regular homologous recombination techniques to make it what's called a knockout mouse or a gene replacement mouse. And as shown here, this is basically how it goes. So in the first step, you have to have a source of mouse ES cells. So this is like, you have to get some ES cells and they have to be growing in the lab. And then what you can do is that originally these would be derived from a blastocyst mouse embryo, as shown here. And you can see them in green growing in the Petri dish there. And then the other component is that someone in the lab has to engineer a targeting vector. And so sort of the nuts and bolts of this can happen in the computer, and it can be designed in the computer, but ultimately someone will have to go in and do the cloning to make this piece of DNA. So by the cloning, I mean to perhaps amplify up certain sections, cut and paste them together. These days, you can outsource a lot of that kind of work, or sometimes you can just have DNA synthesized by a company. But it's really important to construct the targeting vector, which is shown here on the right. The key elements of the targeting vector are that it has the piece of DNA that you wish to be inserted. So in this case, this is highlighted in the red and the blue, shown here. And then there's this homologous DNA, which you've engineered to be complementary to the target site that you want to target. And this is super important because you don't want the targeting vector to insert into the host genome, in this case the mouse, in a random place. You want to tell it exactly where to go because, for instance, you want to exactly remove the activity of a specific gene, right? And so the homologous sections are really what impart that specificity into the process. Okay, so there are many ways to introduce this DNA construct that includes the targeting elements. And so here we just say transfection. So you basically get it into the cells. And then once it's in the cells, it finds its target gene. And you can see here that shown on the bottom right is a very similar cartoon to what I showed you from that textbook earlier. And this is the spot where homologous recombination is happening. And what you want to notice here is that in this case, we're basically inserting this gene that's depicted in red. And in this particular example, it confers resistance to a specific antibiotic. And so that's how you can find those red cells in the huge population of cells that are not targeted. So, you know, if this is a very lowly efficient process, which it sometimes can be, then you basically want to find that needle in a haystack. And by introducing this particular antibiotic resistance gene, it gives you a tool to select for those. So you can grow these this heterogeneous mix of targeted and non-targeted cells in culture in the lab, and then you can give them an antibiotic that will either kill all the ones that don't have the insertion, or select or positively select for the ones that do have the insertion. There's two ways to do it. Okay, so that's kind of a detail. And what happens next, right? So now you have some cells, and those are mouse cells, but they have an engineered version of a specific gene, and so you want to do some screening to confirm that. And then once you've confirmed it in these cell cultures, then you can take some of those cells and you take a new blastocyst mouse embryo shown here. So there's another donor animal that donates some blastocyst embryos, and you can see that the experimenter is injecting those little edited cells straight into the blastocyst. And once they get in there, they intermingle with, let's say, the presumably wild type cells. And so now there's some yellow cells and some red cells. And these are called mosaic embryos. And we'll get back to that later because this is a common feature of genetically modified embryos, especially in the first generation. They're called mosaic because some cells are wild type, whereas some cells have the engineered mutation. And so then you need a mouse who's going to be the surrogate mother for these embryos, and you implant usually many of them into this mouse, and then she gives birth to a bunch of mosaic baby mice, and these are shown here. And they have various amounts of edited cells and their progeny. So they create these clones whereby all of the red cells give rise to many daughter cells, but so do the yellow ones. And so basically what you're looking for are ones where lots of the cells are derived from the edited allele of the gene of interest. And you can infer some of this by coat color, which we're not going to get into right now, but they carry markers for coat color also. And so you can kind of pick the ones that are most likely to be the ones you want. And so then the next thing that happens is that you take some of those mosaic mice and then you breed them to a wild type normal mouse. And then eventually you can establish these lines in which all the cells in the body of the mouse are targeted. And these are called the knockout mice if you're looking to inactivate the gene. But again, you can make other kinds of mice, like you can, instead of having a loss of functional allele or just knocking out that gene, you can kind of try to mimic the particular human mutation of interest, and you can make mice with those mutations. So this is an incredibly powerful technology that has been used in the past several decades to great effect to really figure out the mechanisms of human disease, for example, amongst other things. Okay, so that method's been around for quite some time, and it's been used not just in mice, it's been used in many different species. You name it, like many species have been successfully edited using homologous recombination. So it's been quite powerful. But there are some limitations to the homologous recombination method, and there is this urge to want to make things like even simpler, right? And so if your whole goal is really just to disrupt the gene, let's just pretend, one thing that you could do is just to sort of get an element into the genome that goes and messes with that spot in some way that the cell isn't going to correct it normally, and then you can screen for these mutations. And so this is actually quite a bit less labor intensive because you can sort of pre-buy many of the parts and have them synthesized, and you can basically shortcut much of what I just told you. So where does this technology come from? It has its origins in actual biology, and the kind of biology it has its origins in is shown here. And so in gray is basically a portion of a bacterial cell. And in blue is a DNA virus. So these are really tiny viruses that infect bacteria. And you can see that what is being shown here is that the DNA virus typically is being extruded into the bacterial cell. And bacteria, even though they don't have an immune system the way that we think about how humans have immune systems, they do have immune systems in that they have responses to things that pose a threat, like a virus. And so bacteria have evolved to have these things shown here called CRISPR. So this is the CRISPR locus in a particular bacterial genome, just a generic simplification here. And what you can see is that at some point, some of the viral DNA can get integrated. And then what happens next is that this stimulates the cell to do something. And so basically, not only is that viral DNA now being transcribed into viral RNA shown here in the middle, but also RNA is transcribed from the other elements of the CRISPR locus, processed and then bound to this thing in green, which is called the Cas9 protein. So this is a really important enzyme, and it's sort of presenting all of these short pieces. Some of them are endogenous to the bacterial genome, but some of them are also the viral genome. And seeing that the viral genome is there is enough to stimulate this third step, in which one of these small RNA molecules complexed with this Cas9 protein seeks out and destroys the viral sequences. And so this is a way that sort of the bacteria have outsmarted the virus. So what it's doing here is it's pairing with that same element up in the viral genome, the new viral genome from a new virion that's infecting it. And so the next thing that happens is that it cleaves that viral genome of the second virus that infects the cell and renders it incapable of doing the naughty deeds that viruses do. Okay, so how do people use this to engineer genomes or modulate gene expression in other organisms? What's really cool is that Cas9 protein that I showed you in green can be programmed by the researcher to carry a specific guide RNA with homology to the DNA sequence of interest in the genome you want to modify. The only thing that you really have to have is something called a PAM sequence, and that is just a short sequence of DNA that is several bases next to the target site. So instead of going and attacking the viral genome and making little cuts there, it's going to attack the genome of the species that you're trying to engineer, which could be a butterfly or a salamander or whatever. You just need the enzyme and then a guide RNA, and then you need them to be ejected or in some other way transfected into a cell, and then they can go and get together and basically make a cut in the gene of interest, which is encoded by that guide RNA again. And then it's up to the cell to figure out how to either fix that or kind of fix it. And by kind of fixing it, I mean the cell can use any of these repair pathways after this cut happens. And some of them are sloppy, and some of them will end up giving you exactly what you want, which is, in the case we're talking about now, a loss of function mutation in the gene. So it's your job as the researcher to figure out which cells carry that loss of function gene and which ones don't, but there are methods to do this, which we'll talk a little bit about. Okay, so once the Cas9 enzyme makes this double-stranded break in the target, several things can happen. It can either get repaired by non-homologous end joining, which is on the left-hand side of that slide I showed you earlier, or by homologous recombination. But either way, it's possible that a mistake can be made, and that could become a mutation. And it's also true that you can use the same kind of methodology, but adding a couple more bells and whistles to not only break a gene, but basically to break a gene and stick something specific in there as well. Okay, so what does this look like? Here we have the Cas9 protein that I showed you, and then shown in sort of like this mustard color is the double-stranded DNA in the genome. So if in my lab, this would be like the salamander gene locus that we're interested in editing. And the little yellow thing is a PAM site. So that's going to be a site that's endogenous to the host genome, but is recognized by the Cas9 complex. And then you can see the user provides this blue thing, which is the guide RNA. And typically, you and your students would design these, and there's some design principles that I'm sure you'll get into with LabXchange, but you design these and they should be quite specific for that locus and nowhere else in the genome. And then when you get the protein and the guide RNA into the cells of interest, then they can, the guide RNA can base pair with the sequence you want to edit, and then the Cas9 enzyme can cut it there. Okay, so that's basically how it works. These are kind of more details than you probably need at this point. And the long story short is it can make a double strand break, and that how the cell decides to take care of that will either result in a perfect repair, i.e. no mutation, or a mutation. And the mutation can be deletion of some bases, insertion of some bases. If it's deletion, it can be deletion of 2 bases, which could cause a frame shift and then a premature stop codon, etc. So really what you want to do is get in there and sequence these cells and figure out, who's got what kind of mutation, and then use that to inform the next stage of your biology experiment. Okay, so how was CRISPR first exploited to make targeted changes to eukaryotic genomes? This stuff has not been around in scientific use for a very long time. So if you look, this paper, which was quite foundational, was published in 2013. And so what we can see here is that we're going to be looking at cells, they're human cells, and the locus that they wanted to edit is called EMX1. And you can see that basically the guide RNA will base pair with a sequence within something called a protospacer. This is super important because it's next to the PAM site. And we saw these in the earlier slide. Okay, so here's the little PAM site shown in magenta, and then there's the endogenous locus, and then sometimes this is called the CRRNA or the cryRNA, and then it's also called the guide RNA, the gRNA. That's what we usually call it. So you can see that it's binding here. And then the next thing that happens is that it goes and makes mutation. If I were teaching the undergraduates, I would say, well, what kinds of tests would you want to do to see if the editing works the way you'd hope? And we would talk about that. You might be doing this in your classroom. If you are doing it in the classroom, there's basically 2 common ways to figure this out. And in the first way, it doesn't rely on sequencing technology at all. It basically relies on the fact that if all of the species in a population of cells is wild type, then when you melt those nucleic acids at that locus apart, so all the DNA apart, then when it gets back together, then they're going to have a 1-to-1 match, because the strands are exactly the same, right? There's no mutation. And if everything had a mutation, those would also have a 1-to-1 match because they'd be exactly the same. But if you have induced some mutations in a population of DNA, then what will happen when you melt the whole population and then you allow them to re-anneal these DNA molecules, if there's a mutation in one and the other one is wild type, then they're going to be zipped together everywhere except that little spot where the mutation is. And that little spot where the mutation is either going to make it's going to make a bulge probably, or it's not going to base pair correctly. And that is the signal for a particular enzyme that can go in and specifically cut these heterodimers of DNA. So again, if the whole population is wild type and there's no editing, it's not going to cut. If the whole population is 100% mutant in the exact same way, which is highly unlikely, then it also won't cut. But if you get these hybrid molecules that have a mutant version paired with a wild type version, then there'll be a little kink and this enzyme will cut it. And how do you resolve that? Like, how do you detect that? Here we have a gel. And in this case, what the person has probably done is PCR amplified the region of interest, which is 684 base pairs. So you see that big furry band at the top. PCR worked well. And then after that, what they've done is subjected the PCR products or the amplicons to some kind of digestion with a particular enzyme that will detect these heteroduplexes. And you can see here that basically all I want you to see is that on the right-hand side, that if you do have both the Cas9 and the guide RNA in this particular enzyme, as well as the particular locus, that instead of having 100% 684 base pair amplicons, some of them get cut into these smaller pieces, which if you add them together, it's 684 bases. And that's just a measure of how, those little bands show you that those are the pieces that were cut at the spot where these mutations were made. And so this is awesomely impressive in some ways, because this is the first time that it was demonstrated that human cells can be edited in this way using CRISPR-Cas9. But you see the efficiency is not crazy high. It's 4.7% and 5%. And so it's still possible to do subsequent things and enrich for the ones that are mutant later, but a lot of what's actually done in practice in the lab is trying to get that first step optimized so that when you're looking at mosaic animals, more than 5% of the cells are actually edited, for example. Now, another way to do this is to just sequence. And so this is shown over here. And this probably over the long haul takes less effort, and it can be much more multiplexed, for example. And so what we have here is the endogenous locus shown at top with the sequencing read. And then this is shown at the top of that little alignment down there where the wild type allele is at the top. And then there are about 6 different iterations of different kinds of mutations that were introduced. And so you can see that in these mutations, a couple of them are, some of them are missing 2 bases, which would eventually lead to a mess up protein. Some of them are, if you're missing 3 or 6, then for example, you might just be missing 1 or 2 amino acids. And so you can go in and really say, which are the kinds of alleles we want, and then continue on with those particular ones. Okay, so what do you also want to do to make sure that what you created is what you think you created? So besides sequencing, you also want to, if you have the option, take a look at what kind of protein does or does not get made from that locus, right? Because most of biology operates at the level of proteins, and so it's great to have that readout that says, oh, okay, I think I have this mutation, but it would be even better if you could also demonstrate that when you, for example, in a diploid, have knocked out both copies of the gene, that you don't see the protein, right? And so to do that, basically what most people do is they try to get an antibody to the protein of interest and then use something like a Western blot to detect it. And this is just one example here, but this is commonly done in the lab to sort of, prove. And so here we have 4 different proteins, and you can see that these were targeted in different ways, and that for all of them there's an antibody, and also like you might want to try 2 antibodies for any given protein, and you can see that over here. And what I'm trying to show you here also is that sometimes you can get surprised in that insofar as cells do weird things, and we don't 100% understand all of the different ways that they could choose to make a functional protein anyway, or to make a protein that's recognized by the antibody. It is possible that they can start, they can change the frame, or they can have an alternative start site. Examples exist of this. And so what you really want to see is that for the gene of interest, that there's a blank well where your antibody detects the protein. So if we're looking at this Rictor protein shown up here, and if you look at these cells are plotted based on what was the intended target to be knocked out. So here's Rictor, and then here's 3 other genes. And you can see that the Rictor antibody, the anti-Rictor protein antibody, recognizes Rictor in all 3 of the other ones. Well, that makes sense because we didn't try to edit those out, right? But over here, you can see that for the knockout cells, it's blank for there. And so that's sort of like the dream scenario. But you can see that sometimes here we have VPS35, and what they saw here was a very faint band. So there must be some biological explanation for that. But with antibody 2, it was completely gone. And then we look up here. So some of this, what I'm telling you, is a little bit of details, but it's always important to validate, right? I like this one with the asterisk because this shows that in this topoisomerase knockout that they tried to make, when they ran the antibody for it, one of them, it showed that it looked like it was completely knocked out, but the other antibody revealed that the cells are actually making a version, but it's a shorter and smaller version, so it migrates faster in this protein gel of that particular protein. Okay, so these are all ways to validate that you have what you think you have, and then you can go on to use these cells to study the function, for example, of any of these proteins. Okay. So what kinds of experiments has CRISPR enabled? So now I'm going to talk about some of the fun stuff, right? This is sort of like the lab stuff about how you get to the point of doing fun experiments, right? What kinds of experiments has this technology enabled? So shown at left, just within the last about year or so, people have been able to make knockout lizards, and these are really cool looking here. So you can see the animal on the right is the wild type version, which is like different colors of brown and stripes, for example. And then on the left is the mutant version. And what they've targeted here is a gene that is often targeted as a control when establishing a knockout strategy in a new species. And this is the TYR gene, which is short for tyrosinase. And tyrosinase is an enzyme that's required to make pigmentation in many animals. And so if you make a knockout and you delete the functional activity of the tyrosinase copies in an animal and all the cells, then you can get something that looks like this, which is lacking pigmentation. So that is super cool. You know, you can imagine it could be interesting for hobbyists, but more importantly, it's very interesting for people who study lizards in the lab, and there's so many reasons to study lizards. So shown on the right, we have another example, and this was out of China, and here this man is holding something that he calls a micro pig, and it's a very cute pig that has altered proportions of its particular features, body features like limbs, for example, and it just overall develops much smaller. I forget what it was, what they targeted, but I think it was a particular growth hormone pathway shown here. And there could be reasons to do this. Of course, they mentioned like these make cute pets, but they also facilitate really important biomedical research, because in many ways, for many types of research, pigs are actually a really good model for humans. But they're hard to keep in large numbers because they're so large, and they're at like academic medical complexes. And so this could enable more research using the pig model than was previously possible. Okay, so shown at bottom are some salamanders. So these are a kind of salamander called a newt. And what is interesting here, and I'm going to talk a little bit about the salamanders we study next, but what's interesting here is that the field of limb development is much more sophisticated at this point than the field of limb regeneration. And that's because of chicken and mouse primarily, which have served as really excellent models for understanding how limbs develop during embryogenesis. And for the past several decades, this has been really interrogated at a molecular level. And suffice it to say, we know a lot of the genes that are required to make normal limbs, but also when and where they're required, so like what kinds of cells and at which stage of development. And a large question in regenerative biology, so in this example, in how limbs can regenerate, is how similar is regeneration to development? Is it just a recapitulation of development, only on a different scale and on command, like in response to an injury? Or are there more nuanced differences between the two processes? And so what this CRISPR technology has really enabled recently is for people to take that wealth of information about limb development and really ask, okay, well, if we get rid of these particular genes during limb regeneration, what happens? In doing so, you're able to ask, are those genes also required for limb regeneration? And so shown on the left are three different genotypes of these newts. And you can see that the wild type has 2 forelimbs and 2 hind limbs, and each one of them has a characteristic number of digits. And then on the in the middle and on the right are these HOX13 crispit newts. And so this means that they've deleted particular HOX gene alleles. So HOX genes are really important for the development of many parts of the body, and they're especially critical for establishing body axes, and they're very highly evolutionarily conserved. And all I want you to see here is that basically what they observed was that their clue from limb development turned out to be true later in limb regeneration. So these animals developed weird limbs, shown here, but then these altered morphologies were recapitulated on the bottom right when those limbs were also amputated. So you can see that by 39 days post-amputation, the top one looks totally normal. It's got 4 digits, but at 39 days post-amputation, if you're missing either HOX A13, C13, or D13, then you can get something that basically truncates before you get to the hand, or even if you just have missing 2 of them alone. So this is quite interesting from the standpoint of comparing development to regeneration. It's also really cool that now there's finally a tool in regeneration to go into these highly regenerative organisms like salamanders and really study at a molecular level, what are these particular genes doing? So in my lab, we study a different kind of salamander. This salamander is shown here. This is actually a naturally occurring allele of these salamanders that produces this phenotype of, it's called the white mutant. In nature, these salamanders are native to a lake outside Mexico City, and they're critically endangered, very critically endangered in the wild now. But all of the lab populations are derived from a population that came into the lab about 150 years ago. In the wild, they're actually kind of like a mottled brownish black color. So what I want you to see here, hopefully this plays, is that this is an animal from our colony that went to be videotaped over the course of 8 weeks after its forelimb had been amputated. And so this is a time-lapse series of this animal regenerating its limb, and you can see it here. I'm just going to point this bump out right here.
So it lost its leg, but within 2 months, it's going to look virtually indistinguishable from the leg that was once there.
So we're really interested.
Miraculous, and it would be amazing to know how this is going on.
We're really interested in how this happens, and specifically, how do they make that little bump? That little bump is called a blastema, and it's absolutely required for limb regeneration. And the very intriguing thing from our standpoint is that mammals don't usually make blastemas. And so we want to know, where do the cells come from that make the blastema, and what are the signals that tell those cells to undergo this transformation from perhaps like an activated progenitor cell into a blastema cell. This overall process is schematized here. I don't want you to read all the details of this, but all I want you to see is that the blastema is this little bump of progenitor cells, and it's colored blue here. So it's really important that these cells proliferate a lot, and it's also important that while they're proliferating a lot, they don't undergo DNA damage during that process, which was the subject of this study that we had done recently. We had found this gene called ISABSINT2 in the process of studying what genes get turned on during normal limb regeneration and which genes don't get induced in cases of compromised limb regeneration. And this gene ISABSINT2 is one of these genes. It's actually quite interesting to think about that it's named ISABSINT because it was first studied in Drosophila, and in flies only have one version of this, just ISABSINT, but vertebrates have 4, 1, 2, 3, and 4. And in flies, if you have a knockout of this gene, then the flies don't have any eyes, or they have very small eyes. So that's where it's got its name from. And so what we've done here is that we've edited these axolotl salamanders using a guide RNA targeted to ISABSINT2, which was, important to pick out ones that would basically target ISABSINT2 as opposed to 1, 3, or 4. And then in the F0, which is shown here at top, we took those mosaic animals and we analyzed the bulk of the cells derived from them, and we're able to assign a percentage editing efficiency shown here. So 30% or 20%, which is fairly common for some genes, but it was good enough to go ahead and mate them together, and then you can make offspring that have these 2 lesions. So basically they don't have any functional copy of ISABSINT2 gene. And then you can use this to study things like limb regeneration. So what do these animals look like? They look like this. So shown at the left is a juvenile version of an axolotl that's wild type, and you can see that it has 2 forelimbs with 4 digits each. And then shown at right is the ISABSINT2 mutant. The important thing for us is that they did grow normal limbs, and we did a lot of analysis inside those limbs to prove that. And then they live long enough for us to have one chance to study limb regeneration in them. The reason why they die early is because they also have dramatic kidney defects, and this is partially responsible for what's going on here with this sort of balloon-like phenotype, and so they have a lot of edema and swelling. And so if you baby them, they can live long enough to study the process of limb regeneration. Okay, so what did we find when we took these ISABSINT2 mutants and we studied the process of limb regeneration in them? In a nutshell, what we found was that the wild type animals shown at the top at 6 days post-amputation have a pretty big blastema shown here. It's like this little bump distal to the arrow. Whereas the mutants often weren't growing a very big blastema at all, and this was statistically significant. And what we decided to do next was to ask the question, what is the mechanism whereby their blastemas are growing more slowly? And so in this experiment, we took the wild type blastemas from these animals and we compared them to the animals who had the ISABSINT2 gene knocked out. And what we're showing here is the percentage of cells that are actively synthesizing DNA, meaning they're undergoing cell cycle progression or they're dividing. And you can see that this fraction is diminished in the animals that are missing the ISABSINT2 activity. We also use some fancier new techniques to basically take and ask the question, what are the changes in global gene expression in the ISABSINT2 mutant blastema cells versus the wild type blastema cells? And I don't want you to look at all the details here because the take home message is simple. And that is that there were major transcriptional differences between the wild type and the ISABSINT2 mutant limbs. and that many of these differences pointed to a difference in controlling the cell cycle, or how the cells decided when to keep dividing. And this was interesting to us because we know from many previous works from other scientists that DNA damage response is often coupled to cell cycle progression. And so from this broad RNA-seq study and the results of that, we were able to postulate that we might be able to get a chemical inhibitor of one of the other cell cycle genes or cell cycle proteins and recapitulate the phenotype that we saw with the ISAPS-2 mutant animals. And so here we can see that we have this chemical inhibitor to checkpoint kinase 1 and 2. And when you put this in the water of the animals, you can pretty severely block limb regeneration, as shown here. So we're just mapping the length of the blastema. In fact, some of these animals didn't regrow limbs at all. So this was a very sort of cool and early application of the CRISPR technology in our lab. We've used it for several other things as well. Okay, so we were able to connect this phenotype to too little proliferation, just like in the mutant, and too much cell death. Now I just want to shift gears really quick to tell you about some of the really cool applications that other people have used for studying other species using CRISPR. So this one I love because it's making CRISPRed ants for studying behavior. Ants are famous for their social behaviors, but also for their ability to lay down a track and really to find food and then tell the other animals where it's at. And this particular gene encodes a 7-pass transmembrane protein, and it's needed by all olfactory receptors to work. So we know that olfaction is important in these processes of navigation, for example, and telling other ants where food is. And so you can see here that they use CRISPR to make these ants, and this is an ant brain shown here. And you can see the antennal lobe of the brain, so is outlined, which is like where the information from the antennae go. And you can see it's got a certain size here, but in the orco-mutants, it's also smaller. And so when we look at these ant behaviors, how do they compare to wild type, and what might that tell you about their social behaviors? So what we see here is that, side note, ants don't like the smell of Sharpie markers. And so if you make a fresh Sharpie grid on something like a piece of paper, then and you plop an ant in the middle, it tends to just be confined by the Sharpie markers shown here, if it's a normal ant. But if it's an orco-mutant ant, it really doesn't tend to be confined by those Sharpie lines at all. And that's because it can't smell them. And it makes these kinds of more random paths shown here. So I think this is quite cool. But when you also look at what this means for its interactions with other ants, this is displayed here. You can see these are traces over time. If you plop a bunch of ants into a dish, for example, and normally the wild type ones kind of form these chains and follow each other around, whereas the mutant orco ants really don't. And so you can imagine that this is one way in which people could definitively say that gene is important for these kinds of social behaviors and also for these olfactory avoidance behaviors in ants. Okay, what about humans? I'm not going to get this much into this, but this has made a lot of news, and so your students may ask about it. So here is the guy from China that made the edited human beings, and there were 2 babies born, and the target was the CCR5 gene, which is required for HIV infection. There were problems from the get-go about permissions and regulations and regulatory boards, et cetera, about this study. And there are a lot of problems about the study. Anyway, so this is in the news. And so you probably want to take a look at this because if you're teaching about CRISPR, it may come up. But very many scientific ethics premises were violated in the creation of these particular babies. What about the things that are sort of more planned out, longer term mainstream? So shown here is an individual, a human, who's afflicted with a particular disease that we're going to get into in a second, muscular dystrophy. It's a terrible disease, and there's not a lot of great treatments for this. Shown at the right is a beagle. Beagles are one of the main animals in which muscular dystrophy is modeled, and I'll show you in a second what's been done there. So in this case, the target is the DMD gene, and mutations in the DMD gene are what cause muscular dystrophy, or some types of it. And so what they've been able to do is to do somatic editing in the dog and establish it as a preclinical dog model and publish it, which we'll talk about in a second. So what happens in the human? Here are some of the stats, 1 in 3,600 boys. It leads to a variety of really bad consequences. By 10 years old, most patients can't walk anymore. And these are really linked to this particular protein called dystrophin. And it's a very highly repetitive protein, and its job in the cells is to link the cell membrane to the cytoskeleton. It sort of acts as a shock absorber. If cells don't have functional copies of dystrophin, then the muscle cells are easily injured and very fragile. Several labs published the CRISPR editing of a mouse model of DMD in 2016. Okay, so basically what I want to show you here is that I said that there's like a lot of repetitive copies of this gene, and the humans that have mutant alleles of this gene often will have this particular mutation called delta EX50, which basically means that you're missing this exon, and then this leads to messed up protein downstream of that exon. And so what they've done here is to model this in a dog, and you can see over here that the way that they're getting the editing ingredients into the dog is by a viral approach. And so basically they're infecting the cells with a lentiviral vector that will go in and release, it encodes the Cas9 protein, but also brings the guide RNA with it. And so what we want to look at here is that, again, here we have a Western blot. So we're detecting the protein, which is great because that's the species, that's the mode of action, right? And so we see in untreated animals that are totally wild type dogs that there's this DMD protein, and it's about this big, and it's right here. But in the dogs that have that mutation, the delta X50 mutation, there's no detectable normal DMD protein shown here. And that if these are controls, so these are the sides of the dog's limbs, like the left limb, let's say, that didn't get the ingredients to do the editing, and so they're also basically not producing any protein. But in the ones that got the adenovirus, so this is an adenovirus delivery, sorry, I just told you it was a lenti, but it was adeno. So an adenovirus with all of these ingredients, then basically you can create a genetic rescue that leads to functional protein shown here. And that will be what matters for the human patients, because they just need the functional protein, right? And if you dig this reference out, it's really quite beautiful, because here what they've shown is even better than that Western blot. So this is, these are sections through the muscle of these animals. And what I want you to see here is that normally the muscles are bundled up in these beautiful arrays shown here. Depending on where you're pulling them from, they have sort of different amounts of packing. And you can see here in the untreated animals that there's a wasting of the muscle, basically. However, in the animals that got one dose of this AAV virus that encoded the editing ingredients, you can see that a decent number of the muscles appear rescued in their morphology. And so they look more wild type. And if these animals were given 10 times that amount of virus, or almost maybe 5 times that amount of virus, then what you get is something that looks really pretty close to wild type. Ultimately, it'll be really great to see what the function is, you know, for example, of that muscle in the freely behaving dog. And then you would also want to think about, well, what kinds of data would you want to see before your institution, for example, would grant permission for someone to test this in people, or your company, for example. So this will all have to be thought out, because there's a lot of important ethical implications. Okay, so briefly, I just want to highlight some other possible uses. We know that there are way more people sitting on the organ transplant list than there are available organs. And so this has been something that's been postulated for a very long time, is would it be possible to harvest organs from another species and use them in a human? And the pig is often brought into that conversation because of many aspects of the pig that are similar to humans. However, there's a problem with that. And that is that when some individual cells or like very small tissues have been transplanted into humans from pigs, they don't live very long. Those cells end up getting cleared by the body, and the body has basically mounts an immune attack to them. And so this has to be overcome in order for this to be a viable solution. Immune cells are really programmed to recognize non-self cells and will go in and mount an attack. And then the thing I wanted to talk about today is this other thing called PERVs. So PERVs are endogenous retroviral elements. And so what's interesting about this is that, you know, all species have these sort of either active or vestigial transposable elements, so pieces of DNA lying around in their genome. And these can be a source of incompatibility between species. And so what's been postulated is that if we can get rid of these in the pig cells, and they would make better donors, right? We don't really have time to get into all of this, but if you want to talk about this later or after class, or we can do this too. But there are other ways that you can also make the human patient a little bit better at receiving donor pig organs, for example. And before CRISPR, back when people could really only do that kind of homologous recombination I showed you with the mouse in the very beginning, maybe like 50 or so, maybe less, maybe at least several dozen genes have been manipulated for the intent and purpose of making modified pigs that would be better donors for humans. And some of these are shown here. But most recently, CRISPR has been applied to this idea about these PERV retroviral elements. So pigs have about 60 of them, and the thought is if they could be deleted or edited out, that these would make the pig cells less immunogenic to the human. And so this is the first pig for which that was done. And don't pay attention to all of the little nitty-gritty parts down here. All I want you to see is that when her cells are sampled, they're almost all 100% edited. So this is a map of the non-homologous end joining, meaning edited percentage at that locus from cells derived from these different tissue types in the pig. So this is pretty cool. But what else might you use it for? So this is probably one of the most interesting examples of what it can be used for, and that is this thing called a gene drive. So what is a gene drive? If we just take one step back and we look at these two insects, if we were to genetically modify one of them and alter it, or if this was just a naturally occurring mutation that arose spontaneously in a population, then what might happen in order for it to get passed on generation to generation is that the altered allele is shown in green here, and in this normally diploid organism, if you just follow regular Mendelian genetics, if you're a heterozygote, then there's a 50-50 chance of passing it on, right? Because that's how Mendelian genetics works. However, the researcher can kind of hijack this system of genetics by making something called a gene drive. And in a gene drive, you're not just editing that locus and like messing it up or changing it or deleting it, but you're also inserting the elements that will let this allele on this chromosome go and do it to the matching allele on the other chromosome, right? And so not only did you muck with the allele on the original one, but it's also now got these transgenic elements encoded that encode the Cas9, which is required to cut, so the protein, as well as the guide RNA. And so it can go and basically convert the normal allele in the cell, and in the same generation, change it into the mutant allele. And so what's shown here is that over several generations, this can allow you to engineer an entire population of species, even if this is not something that is really good for that particular species, into this altered version that you might want instead. Okay, so we can think about this as a way to subvert evolution. And let's just pretend like if you had a problem with bunny rabbits and you wanted to eradicate bunny rabbits, even though some people really love them, it can actually end up having really deleterious consequences to the food chain and the environment. And so that's something that students might be interested in thinking about. And that's like, just to imagine, oh, let's pretend that everyone thinks that X is a problem. If we got rid of those things, then what would happen? Like we could say, oh, someone, there was a great white shark on the coast of Maine recently. What if we could just get rid of all the great white sharks? Then what would happen to the food chain? So these are interesting thought exercises. But when we think about something that there's not a lot of love for, which are mosquitoes, even though I love insects, they also vector some pretty insidious diseases. And malaria is a huge problem right now. And you can see here that 228 million cases a year, 405,000 people died from malaria just in 2018. And more than 1,000 kids die every single day from malaria, right? This is terrible disease. It's vectored by mosquitoes, shown here. And there's a really cool and interesting sort of parasite life cycle that goes on with these mosquitoes that vector this particular protozoan shown here. And it's very interesting to think about how much of this has been worked out. But what if we could use CRISPR and gene drive to basically intercept this and stop this life cycle from being able to be completed in the wild? And so people are doing this. They're using gene drive technology to solve this problem. It's not like it's happening outside of the lab right now, but again, it's technically feasible now. And so here what we see are two, in this case, not just insects, but two of these exact kinds of mosquitoes that vector that disease. And what I'm showing you here is that a particular locus, which is required for female fertility, is highlighted. And so if you knock that out, for most genes, you only need one functional copy. Some genes, you need two functional copies. But if you have the heterozygous state, she can still mate, right? But when you make a gene drive, it also carries the CRISPR-Cas9 components, and so it can basically convert the wild type alleles and lead to super-Mendelian inheritance. So in that female's germline, that red mutation can convert the wild type mutation into another red mutation. And then pretty soon, what can happen is the whole population can become null for that gene that's required for female fertility. And then what happens is none of the females can reproduce. And if we just look at these different loci shown here, and they surveyed both males and females, all I want you to see is that there's a very good homing rate. So this isn't just editing, but homing, meaning converting the other allele in this lab experiment. Okay, so what does that look like in the eventually in the wild? Is like if you were to in the lab, this is what happened, and it could happen in the wild if you let these animals go, which is basically that eventually, after just a couple generations, all the females, hardly any of them can reproduce anymore. And you can see down here, we still have one heterozygote shown in the bottom right. Okay, so if it's the only thing that can vector that disease, then you can imagine that maybe this would be a way to get rid of the disease. Again, it comes with very heavy ecological considerations that would have to be made. So the example that I just gave you was breaking a gene, right? I'm going to talk in a second about adding a function back, but real quick, I just wanted to show you that they have taken this out up to 14 generations in the laboratory and shown that the mutant alleles can basically become fixed in the population. So this is a really interesting proof of principle, but again, not happening in the wild yet. Okay, so what I want to leave you on is that even when considering this malaria problem, using these particular parasites and this particular kind of mosquito, there's more than one way to go about using a gene drive to theoretically break the ability of that animal, the mosquito, to vector this disease. And instead of deleting a gene that's required for female fertility, in this case, what they've done is insert two factors that these two are shown here, M2A10 and M1C3. And these are basically like little antibodies that are encoded so that if you're a mosquito and you get this gene drive, then suddenly you're making antibodies against an important part of the parasite. So the parasite is Plasmodium falciparum, and you can see that in this little note I have here is that normally Plasmodium has to use this chitinase, which is an enzyme that's important for helping it chew through the mosquito gut. So if they don't have this enzyme, or if the enzyme is being blocked, then they can't do that really essential step. And so you've broken the parasite pathway there. And so the way that they block it is that they engineer the mosquito to make two antibodies, which are proteins that go and bind up this enzyme. And when the enzyme has a lot of antibodies glommed onto it, it can't do its normal job. And so breaks the parasite life cycle that way. So this is quite interesting and just sort of a complementary way to think about a gene drive. Probably most scientists would say that this is just the tip of the iceberg. People haven't really even been dreaming up all the possible uses for gene drives, but certainly other parasitic cell cycle or life cycles would be possible to consider breaking, and parasites lead to a lot of really terrible human diseases. So expect to see more of this in the future. And the ones that I listed here are three that I pulled from the medical literature for which there are already CRISPR-Cas9 and gene drive technologies being investigated. Okay, so finally, I just want to leave you with that, and I can take any questions. I would prefer to see the people asking me the questions. So maybe we can add that back. But yeah, sure, I will take any questions. Or if you want to move on to LabXchange, I can take questions offline over email.
Wonderful. Well, thank you so much, Jessica, for sharing all of this work. And there have been so many comments and great questions coming up in the chat. So I will pose a few of those to you, and we'll see how many we can get through as time allows. So one question that came up is, how do we specifically target a CRISPR locus and complete DNA sequence?
So is this the first question in the Q&A? What can we do to improve the editing efficiency, or how can we target it?
So we had a number of questions that came up, and you're right, this is from a little bit earlier in your talk, and I think you did speak to this a little bit. So, but I think there was just a question about specificity with this technique.
Right. So this is actually this is an extremely important question. It also speaks to the safety, right? So like in a human or in an axolotl, there's like family members, for examples, of genes, and they can be quite similar in their sequence, right? And maybe only one of them needs to be edited to sort of fix the problem, right? But you don't want to edit the other ones. And so specificity is super important. And so like in an ideal situation, you would have a handle as the experimenter on how many other loci are so incredibly closely related at the genome sequence that your guide might also target them. And then you would constantly be surveilling those other loci to see if they were affected by the guide or not. Like, that's really the only way of getting around it. In the best case scenario, you don't have that problem in the first place. When you're designing your guide RNAs, you would have complete knowledge of the animal's complete genome and complete faith that it is like sequenced and assembled and annotated like completely well. And then you would say, okay, I'm going to choose this guide because it is 100% specific to my locus of interest. But that is not how it really goes in practice for many genes. And so you really have to do your homework, and then you also have to constantly analyze to see what else you've affected. And then ultimately, the gold standard in genetics, especially in model organisms where genetics is quite fast, would be to rescue. So whenever you would make like a fly mutant, even if you're using like an old school technology, you would provide a transgenic element, and you would use some kind of promoter to drive its expression, and you would rescue the phenotype. And so that is like one of the gold standard ways to prove that you've done what you think you've done. But these are really important questions, especially if we're thinking about therapies, right?
Great, thank you for that. And another question that actually sort of, there's one that just came up in the chat, and I think these are a little bit related. So you spoke to the technical differences, I think, here, but someone asked what the difference is between the technique that you've described here and using recombinant DNA. And I think maybe another extension of that question is, maybe when do you decide to use recombinant DNA as a method versus CRISPR?
Right. So, I mean, there's a lot to unpack in that question because there's a lot of technologies I didn't talk about. But so I think one thing you need to back up and ask is, do you want the change to be permanent, right? So like once you've edited the cell's genome, all of its daughter cells will inherit that mutation that you've introduced. And so you have to keep that in mind. It depends on what question you're trying to answer in the laboratory. If you just want a transient expression of a particular factor or transiently disrupt something, you might be better off using one of these other kinds of technologies that, for instance, just takes DNA and like plasmid DNA with this, a promoter that's recognized by the host cell and gets it to be expressed, and then eventually it gets, titrated out by cell division or degrades and goes away. Or you can make them fancier, where you can give them a chemical and then it turns it on. So like these days, a lot is possible. You just really have to decide which to use based on the exact experiment and the exact question you're trying to answer.
And I wonder if that sort of answers another question that had come up here, asking, when you're thinking about malaria, what is the best way to do this? Whether it's, I think the question got cut off. It started to say editing female mosquito fertility. It sounds like that might be a situation where you're looking for more permanent change.
Yeah, I mean, I think the people that are proponents of this are really fantasizing that they'll just get rid of the vector. So by vector, I mean the mosquito, right? And then that particular really terrible parasite won't be able to flourish anymore because that is a required vector. So that's like its obligate vector. But I think the people who are urging caution are worried about, well, what if you remove all the mosquitoes from the food chain? What could happen? Or, people also thinking about like the different ways that adding a transgenic element that spreads around, geographically would change things. I mean, to that, it's hard to argue about. Humans have been intervening with genetics since, crops and animals were domesticated, for example, but this is different, right? The gene drive stuff, it's in a way subverts some of the normal controls that are on evolution. And so it's got to be, thought about harder.
Great. And we had a few technical questions here. One was asking about what we can do to improve the efficiency of CRISPR in editing eukaryotic genes.
Yeah, that's a great question. It's something we struggle with all the time in the lab. So my hand waving suggestion is normally in the lab, we actually use we design 3 different guides, all of which should be extremely specific. And we try all 3 of them. And sometimes we don't know why one works like gangbusters and the other one is crap. So a lot of it is just empirical. And so I would always say that it's good to, I mean, if you're doing this as an exercise in class, it's kind of fun to see like the full spectrum that can arise from it. Right? But one thing you want to think about is if you want to edit the most cells possible, you want to do it as early as possible during development. And so like in my lab, we try to inject these ingredients at the 1 cell stage before the, you know, the fertilized embryo has been divided once, right? And so that means like if the earlier the editing happens, the greater number of cells are going to be edited, right? But you can't control everything. Like it doesn't always get inserted, you know, at stage 1, or else everything would be 100% edited, right? It kind of sometimes lingers around and then gets, you know, the mutation gets fixed later or gets introduced later. So that's one thing you can try to optimize is like the timing of the introduction of the things, the ingredients. Another one would be whether you're introducing the Cas9 protein or the mRNA encoding the Cas9 protein, because you can make your model organism express the Cas9 protein if you give it the mRNA, but that takes time, right, to ultimately translate the protein, et cetera. Whereas if you're just directly injecting the protein with the guide RNA, and if you know, you can get them together beforehand, then you can sort of make that shorter. So that's another thing to think about. Or encoding it, like we saw in these mosquito gene drive examples, where the host genome encodes the Cas9 protein.
Right. And another technical question here for you. So how are extracellular vesicles used for gene editing in the laboratory, and are those preferred?
All right, that's a great question. Actually, I won't say you've completely stumped me here, but you're outside of my area of expertise. So by extracellular vesicles, maybe you're talking about like lipofectamine and these kinds of transfection reagents that have been really popular for just transducing DNA elements. Theoretically can also be transducing CRISPR-Cas9. The examples I showed you today, some of them were with the AAV adenovirus, but other viruses can also be used to do this. You know, there are good pros and cons to using a virus, as there are pros and cons to using extracellular vesicles. The thing that I guess people would like about extracellular vesicles is that, you know, they're kind of ephemeral, right? And so you're not so worried about, you know, that you're introducing like a virus into the equation. But my guess is that for human therapies, and from what I've heard for human therapies, a lot of them are going to be viral vectors, but this doesn't mean that once they get into the cells, those cells are cranking out live viruses. They're usually stops into that process. So they're engineered in a way that they can't go and like, you know, actually produce disease, for example. But these are all important things to think about, right?
Great. And so I want to be mindful of your time. Again, there's lots of questions, but I'm going to give you 2 more if that's okay.
Okay, yeah. You
Great.
also have to do the LabXchange thing, so I totally.
That's all right. We'll get there. So this is a great question. So genetically modified mosquitoes have been introduced to Singapore and Brazil to kill the larvae of mosquitoes that cause dengue. So is there a difference between the gene drive method that you've mentioned and the current technology that we have to create genetically modified mosquitoes?
So you've actually stumped me, because I don't know the exact technology that they're using for that. My guess is that, like you say, it's not a gene driver. There would have been more arguing about it. So I don't know for sure exactly how now I'm going to have to look it up, how those technologies are working. But it is an older idea than the gene drive to think about modifying the vector, you know, the vector animal, and then releasing it and hoping that it outcompetes. I mean, the only thing that from a efficacy standpoint that people would be concerned about there is, well, are we sure that that modification doesn't render them less fit, right? Because then it will just be lost over time, right? In the population of the vectors. But I'm sure the people that are doing this, you know, have thought of that and have investigated it. So this would be really interesting.
Great. And then for the last question, I am guaranteed that this is not going to stump you. So I'd just love to hear a little bit about, what excites you about this work and how you got started in this particular field.
Right. So I got started in my field before you could edit a specific locus in an axolotl. I got started right after people had started making transgenic axolotls, which just means taking a piece of DNA and sticking it in the genome, without specifically knowing where it's going to go, right? But that was sort of like the tip of the iceberg, and it indicated that a lot of the other technologies might be adaptable to axolotls. And so the reason why I wanted to go into this at the time that I did was because people have been wondering for like 250 years how salamanders regenerate limbs, but the exact molecules have been hard to prove because there wasn't that kind of robust genetic technology available to do this kind of work. It's still really hard because they have a 1-year generation time. So flies, it's like 2 weeks, and mice, it's 6 weeks from the time that animal was an embryo itself to when it can have its own offspring. And so it's still incredibly labor intensive to make these mutant axolotls. And I didn't show you all the ones that we made that still regenerate legs perfectly well, even though it seemed like a really good gene to knock out. And so, like the possibility of getting burned is quite high. So you can't really put all your eggs in that one basket, but it is a very cool time to be working on salamanders.
Wonderful. Well, thank you again so much for your time. Just so valuable. We appreciate you joining us today.
Bye. Thank you.